So, good morning, good evening, good afternoon, wherever you are. It's a great pleasure today to introduce the first speaker of the second set of the talks on the neurobiology of addiction, the worldwide seminars on the neurobiology of addiction. So today I'm here with David Belin, who is now a professor in psychology in Cambridge. He started as a molecular biologist, actually, and became a neuropharmacologist in the lab of Véronique Doroche-Gamon in Bordeaux, where he established a behavioral model of rodent addiction based on 3 criteria, which is widely used now. He then became a postdoc in 2006 in Barry Everett's lab at Cambridge, went back and into France, and started his own lab in the INSERM. And since 2013, he is now a group leader at Cambridge University, and as I already said initially, a professor in psychology. So David, it's great to have you here today. And without any further ado, the microphone is yours.
Thank you very much, Christian. Thank you to the organizers for inviting me today. And I will try to convey today the notion that if there is no drug addiction without a drug, perhaps the maladaptive nature of drug addiction has to do with something else but the drug. So, as I just said, there's no drug addiction without drugs, and we know that all addictive drugs increase extracellular dopamine in the nucleus accumbens. All addictive drugs, one way or another, influence the mesolimbic system from the ventral tegmental area to the nucleus accumbens. That has been shown in animals using here, for instance, microdialysis, or even in humans in PET imaging studies. And the ability of drugs to increase extracellular dopamine in the nucleus accumbens clearly is involved in their common reinforcing effects, which partly explains why addictive drugs are initially and repeatedly taken. But people do not necessarily initially choose to take drugs for the same reason. Some individuals would take drugs to feel good, to experience novel sensations, and to share them. Other individuals would take drugs to feel better, to lessen anxiety or pain or isolation. And there are a lot of examples of such an effect during the COVID-19 related lockdowns. In overall, they will take drugs to medicate negative internal states. And already, the reasons why individuals take drugs may well substantially influence their subsequent tendency to lose control over drug use and develop addiction. For instance, in a set of experiments, Maxime Prissac in the lab and colleagues have carried out over 7 years, we've demonstrated that animals who are used to live in very rich social environments with a lot of exteroceptive stimulation, when they are placed into the self-administration context, which is a tiny skinner box, they actually come to experience the drug in a state of deprivation of exteroceptive stimulation and social interactions. And while it has been established that indeed animals in such enriched environments tend to take drugs at lower levels than rats in a standard environment, if you were to ask these animals whether they would be willing to work very, very hard to take the drug, whether they would be willing to go through even electric shocks to access the drug, or if they can relinquish their drug responses when the drug is not available, 3 criteria in animals that we have operationalized from the diagnostic criteria of addiction, we've demonstrated that rats that experience the drugs in a deprived state show higher motivation, higher persistence of drug seeking, and higher compulsivity, even though they do not differ in terms of total intake or pain sensitivity from rats in the standard environment. And as a matter of fact, all the animals in that experiment that displayed the full-blown addiction-like behavior characterized by these 3 behaviors came from this enriched environment. These were animals that had engaged in self-administering cocaine, in that case, in a state of social and exteroceptive deprivation. So clearly, the reasons why individuals take drugs may well shape their subsequent vulnerability to addiction. This has been well established in humans as well, and there's even theories about it, the Kantian self-medication hypothesis of addiction, which have long been recognized. And as I just said, addiction is not just about taking drugs. These are shocking figures, but that shows the extent to which individuals will go, and actually the consequences on their bodies or their life individuals will go when they take drugs. So addiction is a compulsive disorder. It is characterized clinically by a set of clinical criteria, such as taking more drug than intended, unsuccessful efforts to cut down the drug, strong urges and craving for the drug, excessive time spent acquiring the drug, activities given up because of use, failure to fulfill major role obligations, and a bunch of criteria that have to do with the compulsive nature, the use despite negative effects, recurrent use in hazardous situations, continued use despite consistent social or interpersonal problems, and adaptations to drugs such as tolerance and withdrawal sign upon cessation of drug use. All these behavioral manifestations tap onto a wide array of brain structures, and they are not restricted to the mesolimbic system. So addiction is a brain disorder. And as I've just mentioned, we are not all equally vulnerable to developing drug addiction. Out of a population of drug-naive individuals, some, actually most of us, will take one drug or another, including alcohol or nicotine. Some individuals will try drugs and they will experience their effects, like them or not, and they could continue, even for a lifetime, to take these drugs recreationally. They maintain control over drug use. Some individuals would actually at one point shift to loss of control over drug use, display this compulsive drug seeking and taking pattern that characterizes addiction, and show a high propensity to relapse. As a matter of fact, work I have initiated and followed by many others now has shown that it's the same in animals. If you take a large cohort of outbred rats, for instance, and Christian have shown the same thing in mice, as well as Veronica Alvarez, some animals would actually differ from the others in the way they display behavioral manifestations that are reminiscent of the compulsive nature of drug addiction. So while most of the animals exposed to drugs in a self-administration setting would take drugs readily, they would maintain control over this use, and only a minority of these individuals would eventually lose control over drug intake. Christian has also shown that it's the case for optogenetic stimulation of dopamine neurons. So clearly, addiction is not just about the effects of drugs on the brain, right? It's beyond understanding the adaptation of the brain to drug exposure. Because if we compare individuals like myself, I do like whiskey and I do drink whiskey, to drug-naive individuals, we will understand the effects of whiskey exposure on my brain. But I hope, perhaps I've got insight deficits, but I hope I'm not an alcoholic. So that would tell us very little about the brain basis of addiction. And it is very important to factor in the interindividual vulnerability to develop the compulsive nature of drug use that characterizes addiction, and compare it to the effects of drugs on the brain of individuals who maintain recreational drug use. And this interindividual vulnerability and the psychological and neural basis of such vulnerability have been the burning question in my lab for quite some years now. So I've just mentioned that clearly addiction is beyond the mesolimbic dopamine system. And there are many evidence that suggest that drugs interfere with many other systems than the mesolimbic dopamine system. And this is, to me, a seminal study from Nora Volkov, published in 2006 in Journal of Neuroscience, in which she showed that individuals with an addiction to cocaine presented with drug-paired cues, they crave in response to these cues, and that craving is associated with an increased dopamine transmission, as measured in PET here, in the very dorsal parts of the striatum. This part of the striatum that, when there's not enough dopamine, you suffer from the motor symptoms of Parkinson's disease and other symptoms, of course. So the motor parts of the striatum. And thereby suggesting that over the course of drug exposure, there is a transition in the neural locus of control from the mesolimbic system that mediates the reinforcing purpose of drugs to other striatal systems that are more to do with motor control and, we'll see, habits. So the same transitions have been shown in the context of alcohol in a series of elegant studies from Volstein Klein, who show that if you take an alcohol user, a recreational user, and you present them with cues, there is activation of the ventral part of the striatum here. And if you present the exact same cues to a heavy drinker, it is the dorsal part of the striatum that is responding to these cues. So once again here, a ventral to dorsal shift. Well, does it matter? Yes, it does quite matter, because these intrastriatal transitions, these shifts from ventral to dorsal striatum, have been reported for every single drug. And importantly, this very elegant study from Cox has shown that these transitions to DLS response to cues, dorsolateral striatum response to cues, occur in ongoing users prior to the onset of drug addiction, in individuals who take cocaine but do not yet meet the diagnosis of cocaine addiction. So it seems that it is perhaps preceding, setting a stage for the establishment of drug addiction. As a matter of fact, we have known a lot about these transitions for a long time, especially seminal studies from Linda Perino, who showed in primates, in looking at 2-deoxyglucose metabolic activity decreases or dopamine D2 binding receptor site decreases, that the decreases in response to drug exposure following weeks or months of self-administration were initially restricted in the ventromedial part of the striatum in primates, and following chronic exposure, here we're talking years, these adaptations have spread throughout the striatum, eventually to encompass the caudate and the anterior putamen. And these alterations in metabolic decreases have been paralleled with decreases in the dopamine D2 receptors from ventromedial to dorsolateral striatal pattern. In rodents as well, similar alterations have been shown by Morgan Besson, with whom I've had the pleasure to work in 2012 and 2013. Morgan used in situ hybridization in rats exposed to 12 days of cocaine self-administration or to 50 days of cocaine self-administration. And she measured the mRNA levels of the dopamine D2 receptor. And she showed that after a short-term exposure to the drug, there were decreases in mRNA levels of the D2 dopamine receptor in the nucleus accumbens shell, this ventral part of the striatum here, whereas following chronic self-administration, the same kind of history, which we had previously shown to be necessary for rats to develop these 3 criteria for addiction-like behavior, after such a prolonged history, then these decreases in D2 mRNA levels were observed throughout the entire striatum, encompassing the nucleus accumbens core, dorsomedial, and dorsolateral striatum. So clearly, there are transitions within striatum, but what does that mean? Well, one needs to come back to a bit of learning theory and understand the assistive mechanisms in drug seeking to address that question. When individuals, or rats or mice, press a lever, they can be pressing that lever for 2 different reasons. They could be pressing this lever because they have a previous knowledge of the value of the outcome of their response and the causal nature of the relationship between their response and the outcome. In other words, the rat presses the lever because they know they will receive cocaine and they have an idea of the value of cocaine or alcohol or heroin. That is goal-directed behavior, a response that is predicated on a representation of the motivational value of the goal. And at the neural systems level, such goal-directed behavior depends on a circuit, which I will brush over very quickly, that depends on the basolateral amygdala, the nucleus accumbens core, and the posterior dorsomedial striatum. But the exact same response can be supported by a different kind of association called a stimulus-response association. And in that case, the animal relies on specific features in the environment. And when I speak of the environment, I speak of both external cues and internal cues, a specific setting that drives a response. And the outcome of that response is the drug. In that case, the animal learns to respond because the setting dictates the response, and the outcome is the drug. Such stimulus response, habitual drug-seeking behavior, depends on a very different neural system that involves the central nucleus of the amygdala and the dorsolateral striatum. So, in other words, the same response can well be mediated by 2 different psychological processes and 2 different brain circuits. So, why is that important? Well, because often it is very difficult to distinguish between these responses. When you look at the assistive nature of goal-directed behavior, the response is preceded by a representation of the relationship between the response and the outcome. The response leads to the outcome being delivered, and the delivery of the outcome, provided the outcome is of the expected value, reinforces the response-outcome association. So the response persists. If you extinguish this behavior, train the animal, and you omit the outcome, the omission of the outcome is going to result in a decrease in the strength of the response-outcome association. Hence, the animal will stop responding. The problem is, if the behavior is mediated by habit, you've got the exact same thing. In a habit, the response leads to the outcome, and the presentation of the outcome reinforces the stimulus-response association, such that each time the setting is presented, the animal is going to respond. So the maintenance of the response depends on the presentation of the outcome. And if you no longer deliver the outcome, well, the outcome is not reinforcing the stimulus-response association anymore. The animal will stop responding. So the associative nature of instrumental performance cannot be measured under reinforcement or extension. Okay? So in that case, it's very important to bear in mind that habits are not skills. Habits are the psychological mechanisms that govern the initiation of a behavioral sequence. They tell you very little about the performance of that sequence, okay? Habits are not skills. And that's quite important because many people tend to confound the two, mistake one for another. So for instance, I can have a goal-directed action outcome overarching schemata. I need to go to the supermarket and I'm going to drive there. And in the performance of that behavioral sequence, I can recruit skills, action outcomes, subroutines, or habits, okay? In the case of addiction, I would suggest that actually the entire behavioral sequence is under a stimulus-response cognitive schemata, whereby I am a drug addict, I wake up, and my interoception, my autobiographical memory, these are cues. These cues set a setting that triggers a specific response. I rationalize this response by stating that I want to get high, but the habitual cognitive schemata has already initiated drug foraging. But within this drug foraging, I can implement skills and I can implement subroutines that are goal-directed. I can have flexibility in the performance of the behavioral sequence. The behavioral sequence is inflexible in the sense that it is initiating overall through a stimulus-response association. So how to determine the assistive nature of instrumental responses then? Well, what you would do is you would train a rat, for instance, for food to respond for food, and eventually you would devalue the outcome, either by pre-feeding satiety, you give the rat as much food as he wants before a session, or with conditioned taste aversion, you make the value of that food very, very low. And then you test the animal under extension, probing the representation the animal has of the value of the outcome of the response. There are two scenarios possible. If the animal decreases its responses for the valued outcome as compared to the devalued outcome as compared to the valued outcome, that shows that the response is dependent on a prior representation of the value of the outcome. And the animal tells you, I'm not going to work for something I don't want. If the animal persists in responding, well, that is the signature that the response is not predicated on a representation of the value of the goal. It is dictated by a setting. And that is important because if you were to then give the animal food for free outside any instrumental setting, well, in both cases, the animal would not eat the food. So when the behavior is under SR control, when the animal doesn't have access to the food, they would respond for it. Or would they respond for the food, or would they respond because it's a habit? But when you give them access to the food, they won't eat it. So you can't really say they want it if when they have it, they don't take it. So it's very important when one discusses habits to measure seeking responses. And that is perhaps the only thing I would like, especially the young researchers among ourselves and the students to remember. There's a big difference between seeking responses and taking responses. And seeking responses are responses expressed prior to the obtention and the consumption of an outcome, but always leading to an outcome. So responding under extension is not a seeking response. In the case of self-administered drugs, Zapata in Shepard's lab did an experiment I'm very jealous of. I wish I had done that experiment. He used the seeking-taking task that had been developed in Cambridge by Barry Ritz and Tony Dickinson and Sarah Olmsted, whereby rats would learn to press on a lever called the seeking lever. And the consequences of these responses are that eventually the rat has access to another lever called the taking lever. And then the rat can press on that lever to receive an infusion of the drug alongside presentation of the drug paired cue. So seeking, taking task. There is a spatial and temporal dissociation between preparatory responses and confirmatory taking responses here. And then what Zapata did was to extinguish the taking lever. So now the animals are trained in the absence of a seeking lever. They respond on the taking lever, and the taking lever no longer gives access to the drug. And you subsequently train the animals on the extinction on the seeking lever, and you ask them whether they know that the seeking responses give access to a taking lever that itself doesn't predict the drug anymore. Does it make sense? So in that case, we devalue the goal of the seeking response, which is the taking lever that gives access to cocaine. And if you do that after 10 days of self-administration, there is a strong effect of the devaluation of the taking lever on seeking responses in these rats, showing that rats early on respond on the seeking lever under a goal-directed control. They know that the responses on the seeking lever give access to the taking lever, which itself leads to cocaine. But following 50 days of self-administration, the animals have become completely resistant to the devaluation of the taking lever. Their seeking responses have become, by definition, habitual. And at that stage, if you infuse lidocaine into the dorsolateral striatum, in other words, you inactivate the dorsolateral striatum, while these infusions have no effect on baseline performance, baseline seeking behavior, they restore sensitivity to the evaluation of the taking lever as compared to vehicle. In other words, it shows that the seeking behavior, when it is habitual, it depends on the dorsolateral striatum. But if you inhibit the dorsolateral striatum, the goal-directed system takes over control of behavior. That reveals, once again, a well-established balance that exists between the goal-directed system and the habit system. One takes over control over behavior as compared to the others, but if one is taken out, the other one resumes control. So Cara Giuliano, following a series of similar studies from Tricia Janak and Lara Corbett, used the seeking, taking, drinking task for alcohol. So in this task, rats are trained on the seeking lever. They seek the drug, eventually to have access to a taking lever. And when they press on that taking lever, now they have access to a bottle of alcohol and they can drink. So it's a seeking response, taking response, confirmatory response chain. And by measuring their sensitivity of these animals to extension of the taking lever, Cara showed that early on the behavior is goal-directed, sensitive to extension of the taking lever, and following months of training, the behavior becomes habitual. It is no longer sensitive to the evaluation of the taking lever. In another experiment, Cara showed that this shift in the sensitivity to manipulation of the value of the taking lever was underpinned by a shift in the neural locus of control over behavior, whereby early on in training, the behavior is not sensitive to manipulations of the dorsolateral striatum, the habit system, but later on in training, the behavior becomes sensitive to manipulations of the habit system. And the way she showed that was to test animals in the seeking, taking, drinking task, while infusing prior to the session a dopamine receptor antagonist into the dorsolateral striatum. And she measured the decrease in seeking responses following blockade of dopamine receptors in the dorsolateral striatum. And as you can see here, in animals with a well-established alcohol-seeking behavior, there is a profound decrease in liver presses on the seeking lever that is dependent on the dose of the dopamine receptor antagonist infused into the dorsolateral striatum. But the taking responses are not influenced. And that's quite important because these two responses are literally pressing a lever. It's the same skill. One is a seeking response that is very distant from the confirmatory response. The other one is a response that one could consider being part of the anterior confirmatory chain of events from pressing the taking lever to drinking alcohol. But importantly, all animals were not equally sensitive to this manipulation of dopamine receptors in the dorsal striatum. And we plotted the reliance on DLS control over behavior by measuring how much the behavior was decreased by the infusion of alpha-frequentixol in the dorsal striatum. And using a cluster analysis, we could identify animals in yellow here whose behavior was very much dependent on the dorsal striatum. In other words, if you block dopamine receptors in the dorsal striatum, their responses were substantially decreased. And some animals showed insensitivity to these manipulations. So clearly here, there are inter-individual differences in the tendency to engage the habit system over a protected history of alcohol-seeking behavior. But importantly, when we challenge the animals to see whether they would be willing to take the risk of receiving shocks in order to access alcohol in these tasks. So in the way Kara did that, she would have 75% of the seeking cycles that would be lever presses on the seeking lever, give access to the taking lever, which gives access to alcohol, and 25% of the cycles, animals would receive shock. Okay? So they would be challenged to respond on the seeking lever, taking the risk of receiving shocks. What's very important here is that the shocks are never contingent with drug exposure. And it's quite important because it's not because you introduce a shock in a procedure that you're measuring compulsivity or punishment. And if you punish the animals, if you run appropriate contingencies and actual punishments, there are marked interindividual differences in the way animals would resist to punishment. So we could identify a good 20% of the animals that would resist to punishment over increasing shock intensities, and animals that would stop seeking their alcohol. So these animals are not willing to take the risk of receiving shocks to get to alcohol. These ones, they are much more so. But if you test these animals 10 months later, the compulsive ones are still compulsive, and the non-compulsive ones are still non-compulsive. So compulsivity is a very stable trait once it is developed, provided it is measured as it should. What was very interesting is now, considering these animals that had engaged the habit system in seeking alcohol, when we challenged them with the risk of receiving punishment, what was revealed is that those animals whose behavior was habitual were much more prone to resist to punishment as compared to the animals who had not engaged their habit system. So the establishment of habitual control over behavior, dorsal striatum dependent control over behavior, predicted an increased tendency to show compulsivity. And as a matter of fact, there was a very, very neat relationship between the reliance on the habit system in these animals and the extent to which they would compulsively seek alcohol. In that study, Kara further showed that when animals were punished and responding under the threat of punishment, those who were compulsive were those who could not disengage their habit system. Many of those animals who had engaged their habit system could disengage it in the face of punishment, and they decreased their responses. Those that persist could not show this relinquishment of the habit system. So clearly, there are marked individual differences in the way individuals engage the habit system, but once they have, they are much more likely to switch to compulsivity. And the expression of compulsivity is due to an inability to disengage this dorsal striatum dependent habit-based behavior. So now you would tell me, yeah, but actually, these are rats. People who suffer from addiction, they are not necessarily locked into a rigid pattern. They show strong motivation, they show good activeness, they show flexibility. So habits have nothing to do with addiction. Well, do they? Do they really show flexibility? Do they really decide not to take drugs? Can they decide not to take drugs if they are not being helped? They may show flexibility in the way they get to their drugs. But what's important is to bear in mind that in the real world, habits are not divorced from Pavlovian cues. And everything I've discussed here is in the context of very neat experimental contingencies whereby we control the presentation of Pavlovian cues and instrumental associations. And I will try to introduce to you the notion of incentive habits, which are something slightly different that emerges from prolonged histories of drug foraging under the control of drug-paired cues, like in real life. In real life, people forage for their drugs, and they often have drug-paired cues that are response-produced that encourage them and support their foraging over prolonged periods of time until they get the drug and take it. So incentive habits, I would suggest, are motivational processes in the amygdala insula system that are aberrantly permeating the dorsal striatum-dependent habit system. And I'll try to show you in the next 15 minutes, I think I still have 15 minutes, Christian, how we came about understanding the process that way. So one, once again, needs to come back a little bit about on learning theory. So when a rat responds for a drug, that's the instrumental association, but often there are a lot of cues in the environment that are not contingent upon the animals responding. These cues can be associated with the delivery of the drug. These are conditioned stimuli. And these cues can be predicting access to the drug. These are occasion setters, discriminative stimuli, like the drug context. And as a matter of fact, these Pavlovian cues, they can interact with instrumental performance. There are two types of interactions: Pavlovian to instrumental transfer, which I will not discuss today, and condition reinforcement, which is what the rest of my talk will be about. What's important is that these cues interact with instrumental responding, irrespective of whether it is supported by a goal-directed association or by a stimulus-response association. And Peter Holland has elegantly shown that actually habits are more sensitive to the influence of Pavlovian cues than goal-directed behaviors. So I've just mentioned the notion of condition reinforcement, and I've mentioned to you that in real life, individuals forage for drugs and their behavior is reinforced by the presentation of drug-paired cues. That is called cue-controlled drug-seeking behavior. And that can be operationalized in animals in second-order schedules of reinforcement. These schedules of reinforcement have been developed at NIDA by Goldberg and Sperman, and in rodents have been developed by Barry Ritz with Athena Marco and Mercedes Arroyo in the early 2000s, if I'm not mistaken, in late 90s. And in these conditions, you can measure the impact of drug-paired cues on instrumental responding drug-seeking behavior over prolonged periods of time. How does that work? Well, we first train the rat to respond for the drug. And then, when the rat has learned to respond for the drug over prolonged periods of time under fixed interval schedules of reinforcement, we introduce the drug-paired cue contingently upon the animal responding. In other words, the animal response would be reinforced by the presentation of the Pavlovian cue, and eventually one response would lead to the drug. So what does it look like behaviorally? You can train rats under a fixed interval 15 minutes of reinforcement, which is the gray hexagons here. And in average, under these conditions, rats would press between 50 and 75 times. But bear in mind that under a fixed interval 15-minute schedule reinforcement, the rule is wait for 15 minutes, and then once you press, you get the drug. And of course, rats, dogs, primates, including ourselves, we don't have this molecular clock that enables us to know when 15 minutes is exactly. So organisms will start responding in anticipation of the end of this interval. And these responses have nothing to do with whether the organism receives the drug or not. They can respond 2,000 times, twice, 100 times, doesn't matter. The rule is that they would have the drug once they press the lever after the 15-minute interval has elapsed. So under a fixed interval 15 schedule of reinforcement, one can measure drug seeking because it always leads to drug delivery in a drug-free state over prolonged periods of time. And there is no opportunity for the animal to have a representation of the relationship between what it does and what it gets. these are ideal conditions to promote habits. Under the second order scheduler reinforcement, what happens is that if you introduce the drug paired cue such that it is now response produced, the drug paired cue is presented contingently upon every 10th liver press. What you can see between the green and the blue here is an increase by more than 100% of the vigor with which the animals engage in responding for the drug. The difference between the gray and the blue is the extent to which the Pavlovian cues exerts condition reinforcing invigoration of responding. The behavior is now controlled by the motivational value of the CS acting as condition reinforcers. So there are a lot of people like Christian who take lasers and transgenic mice and put light into the animal's brain to change their behavior. Well, actually, you can do pretty much the same thing with simply manipulating contingencies in the environment. But as a matter of fact, in humans, if you think light is money, well, humans would work under condition reinforcement for their drug. And I would argue that under these conditions, fixed interval 15 minutes of reinforcement, you can measure habits. And under these conditions, you measure something else. It's a habit that is constantly fueled by the motivational value of the drug paired cue acting as a condition reinforcer. Okay? And I will, for the rest of my talk, compare I515, so the habit condition, to SOR, second order scheduler reinforcement, which is the incentive habit condition. Well, does it matter? You could say, well, it's just a matter of baseline, they respond more. Well, actually, when I first started thinking that something was odd here, is when we realized, when I reproduced data that Luc van der Schoen has produced with Barry before I joined Barry's lab, that if you block dopamine receptors into the dorsal striatum of rats with a prolonged history of cue-controlled drug-taking behavior, you dose-dependently abolish the response. You lose the behavior. Whereas I've told you beforehand that when you inactivate the dorsal striatum in animals that are not trained under conditional reinforcement, what you do is you re-engage the goal-directed system. You do not lose performance at baseline. What you do is to restore sensitivity to outcome evaluation. In these conditions, the balance between the goal-directed system and the habit system is broken. If you disengage the habit system, you lose the behavior. The goal-directed system doesn't take over. So that was the very first hint of evidence that suggested that there was something to this prolonged history of cue-controlled drug-seeking behavior that made the entire process different to a habit. So we investigated whether it was the case that this history of cue-controlled drug-seeking behavior could contribute to some extent to addiction. So these are a series of experiments carried out by Yolanda Pena, Oliver, Michael Pro, and Massim Frisak. So we train rats initially to self-administer cocaine under fixed ratio schedules 1, so continuous reinforcement. Then we train them to seek cocaine under fixed interval schedules of reinforcement of increasing duration. And then animals were trained under either the habit condition, I515, or the incentive habit condition, the second order schedule of reinforcement, whereby every 10 liver presses, they have the presentation of the cue, and they only have the drug if they press once the 15-minute interval has elapsed. As you can see here, the animals never differed in their acquisition of drug self-administration, acquisition of drug seeking. It is only when we introduce conditional reinforcement that the incentive habit animals responded much more than the habit animals. And we tested whether habits, incentive habits, contributed to the high tendency individuals with an addiction show to relapse. So here we forced these animals to be abstinent for 3 days. We maintained them in their home cage. And after 3 days of abstinence, we let them freely seek the drug for 15 minutes, as they would do every day, either under conditional reinforcement or the habit condition. And what we observed was that after abstinence, animals in the habit condition, they responded exactly as they did before abstinence. But animals in the incentive habit condition, they showed a twofold increase in responding following abstinence. Clearly, these animals engage in a more profound tendency to relapse, they're responding twice as much as they've done for weeks at baseline. So when we first got this data, I remember Yolanda telling me, well, that's evidence that withdrawal increases the incentive motivation of the drug. But there are a couple of things that are wrong here. First, in order to undergo incentive evaluation of the drug, following withdrawal, the animals must be self-administering the drug. And this is a drug-free state. Secondly, these animals are responding for few injections a day. It was very unlikely that they underwent withdrawal. Nevertheless, we tested this hypothesis. So what we did was to place the animals for 3 days in their self-administration context, but they could not respond. But every 15 minutes, they received a non-contingent infusion of the drug. So for 3 days, they would be in their home cage, brought to the self-administration context, not respond, receive non-contingent drug infusions, and after 3 days, we give them access to the livers and they can freely seek the drug either under the habit or the incentive habit condition. So here what we've done is we've controlled for any pharmacological withdrawal. And what we observed is that even if they had non-contingent cocaine infusions for 3 days, during which they could not seek the drug, they showed the same increase in responding after abstinence than they did when they didn't have cocaine. And no differences were observed in the habit condition. So clearly here, one could say, oh, so the only difference now is that they lost the opportunity to respond. Is this increase in responding due to the fact that they lost the opportunity to respond and they need to respond when they can? But before we draw that conclusion, some of you may well say, well, wait a minute, non-contingent drug infusions in animals with such an history is very different to self-administered drugs. And that is true. So we tested this hypothesis. So now, for 3 days, animals were brought to their drug-seeking context, and the livers were never available, except they were inserted every 15 minutes, and the animal could press once on the liver to self-administer the drug. So they could self-administer the drug, but not seek it. And after 3 such days, they were given access to the livers at the beginning of the session. They could freely seek cocaine. And what we observed was the exact same increase in responding at relapse. So here what that shows is that incentive habits-mediated relapse occurs when seeking behavior, but not taking behavior is presented. The last important control that one had to make was to be sure that the behavior wasn't driven by a withdrawal from the conditional reinforcer, from the CS. We know baseline performance is very much dependent on the presentation of the CS. But at relapse, in animals who had a history of several months of training to respond for cocaine under the conditional reinforcing properties of the CSs, if we do not present the CS on that day, it has no effect. They still show this potentiated tendency to relapse following forced abstinence. So it's not dependent on CS presentation as relapse. So in other words, this is very different to the well-known incubation of craving process that Grimm and Chairman Wise have discovered. I'll try to wrap up in a couple of minutes. So how do I understand this? Well, under these conditions, whether the behavior is goal-directed or habitual, you have conditioned stimuli that can invigorate the responses. But when the conditioned stimuli are response-produced, they become conditioned reinforcers. They enter an instrumental association of their own. And conditioned reinforcers have been shown to be independent of the value of the drug. These are representations stored in the brain with inherent value. And my suggestion is that the representation, the motivation value of the conditioned reinforcer that is imbued from the outcome can be transferred to the response under well-established cue-controlled drug-seeking behavior in a second-order interdimensional conditioning. What does it mean? It means that the response, can have inherent motivational value. So if you do not have the opportunity to respond, you may want to respond, okay? If that is true, one may suggest that out of a behavior which we know very much relies on the habit system, on the day of this relapse exacerbated response, the behavior should be goal-directed because the animal should respond in order to respond, okay? So in another experiment, we trained rats under the habit condition, the incentive habit condition. They show this exacerbated relapse after abstinence. And we probed the activation of the striatal territories using in situ hybridization against C4s. And while between baseline and relapse, there were no differences in the habit condition between the anterior dorsolateral striatum and the posterior dorsomedial striatum, in incentive habit conditions, while there were no changes in the habit system, that is the anterior dorsolateral striatum, there was a massive increase in activation of CFOs in the goal-directed system that is represented here by the posterior dorsomedial striatum. These are rats who've been responding for more than 60 days, and their performance has been dependent on the DLS for months. And at relapse, there seems to be a recruitment of the goal-directed system. So that shows that they inflexibly engage in responding, but at the neural systems level, there is some flexibility going on. And very likely a transient recruitment of the goal-directed system. So our suggestion is that this instrumental deprivation effect that supports this aberrant engagement at relapse reflects a new goal-directed schemata, the goal of which is responding. If that is true, one should predict causally that performance at baseline is dependent on the ADLS, the habit system, and not on the goal-directed system. But performance as rebound is indeed associated with the transient dependence on the goal-directed system. And we causally tested this hypothesis in a longitudinal study in which animals, after at least 27 days of training under cue-control cocaine-seeking behavior, were challenged in a Latin square design with intra-ADLS or PDMS infusions of alpha-pentaxol or vehicle following abstinence or at baseline. So the same animals were tested either at baseline or following abstinence. As we've shown many times over, under baseline conditions, blockade of dopamine receptors in the habit system, the ADLS, substantially decreased cocaine seeking at baseline, and the same manipulations in the posterior dorsomedial striatum had no influence on responding. So clearly baseline responding here is DLS dependent. In the exact same animals, 3 days later, after 3 days of abstinence, the behavior is no longer dependent on the DLS. Only on that day, it becomes dependent on the goal-directed posterior dorsomedial striatum system. So there's a transient recruitment of the goal-directed system there, which I suggest reflects the engagement of a goal-directed schemata, the goal of which is to respond, because the loss of opportunity to respond during abstinence in these animals with an incentive habit triggers a distress and an urge for action that the animals alleviate by responding. And I believe that would be a way to reconcile the very important role of negative reinforcement and what George Krupp has called hypercafe, which is these negative states following withdrawal, but beyond withdrawal. That shows that responding in the animal with an incentive habit is a drive on its own, and losing the opportunity to respond triggers an exacerbated motivational state, a goal-directed behavior, but the goal is not the drug. So I realized that I've been running out of time. I apologize. The conclusions are that a prolonged history of drug seeking under the control of the conditional reinforcing properties of the drug paired cues results in maladaptive habits that we have called incentive habits. I've tried to show you that incentive habits promote relapse in a way that habits do not. And incentive habits in animals prevented from enacting their habits, there's a recruitment of a new goal-directed schemata, the goal of which is not the drug, but the response. The instrumental deprivation effect, this exacerbation of responding following the loss of opportunity to respond, may result from negative urgency due to the fact that the response is imbued by motivational value. I would like to thank you for your attention, but I would like very much to thank all those who've done these heroic experiments. Maxime Frissac and Michael Pro, Lucia Marty-Prats, Yolanda Pena-Oliver, Cara Giuliano, Eric Ducré, Aude Belin-Rossan, and I would like to thank my friend and my mentor, barrier rate. Thank you.
Thank you so much, David. That's a great presentation. So we're going to switch to the question and answer session. So for those in the audience who would like to ask questions, please do so by using the Q&A tab. And Sema is going to ask the first question.
Hi, David, great talk, super elegant as always. So I know that you're talking a lot about the different roles of the dorsal medial and dorsolateral striatum, but I assume that's where the cell bodies are. Where do you think they're projecting to? That's so important for this behavior.
Absolutely right. The circuit needs to be mapped. And not only downstream, but also upstream. And to be honest with you, we have so far been focusing on upstream. So for instance, we have established that the development of control to the DLS, surprisingly so, depends on the BLA, which doesn't project to the DLS. Okay? And it very much depends on the network whereby the BLA controls the nucleic segment's core and the core devolves control to the DLS. Now, that is for baseline performance. When the neural basis or the neural locus of control shifts here in the case of instrumental deprivation, whether there's a brand new mechanism that mediates the negative affects and the role of this distress onto the two systems, I do not know.
So if I may, just a follow-up question on this. Obviously, as always in the striatum, there is D1 and D2 expressing projection neurons. And you mentioned at the beginning that the D2 downregulation is particularly prominent. Are we talking here about the postsynaptic D2 receptors on those SPNs, or are there other D2 receptors? And how do you sort of reconcile this with the idea of a direct indirect pathway?
So there are two questions there, Christian. First, the decrease in D2 that we showed was on the fixed ratio, was under continuous reinforcement. Nevertheless, you're right. And they were both pre and post-synaptic, because with in situ hybridization, when you target the VTA, you necessarily look at mRNAs of dopamine neurons. And when you target the striatum, you necessarily look at mRNAs in postsynaptic neurons in the striatum, right? Mm-hmm. But they could also be in astrocytes. There are very few, as far as I'm aware, local transduction of D2 receptors in dopamine terminals in the striatum. So we showed both pre and post. In a paper in Nature Communications a couple of years ago, we showed that the reliance on the DLS that is revealed by blocking both D1 and D2 depends on a convergent contribution of D1 and D2. In other words, if you block D1 alone or D2 alone, you have the same effect as if you block both of them. So functionally, irrespective of their downstream outputs, they seem to be converging in supporting DLS control of their behavior.
Okay, Sema.
Okay, there are some questions coming in, so I figured I'd read the first one, which is from someone anonymous, but they want to know what your thoughts are on previous work that's demonstrated that animals will respond for the cue lights and the sounds alone.
That is a very good question. So as a matter of fact, this is called instrumental novelty seeking. You can have a look at one paper of mine in Science in 2008, where we had rats trained to self-administer saline for the first day, and they would press 20 times just to receive the CS and saline. But such responses wear off after one session or two. And you're never talking about 100 or 200 liver presses every day for a month on, with simply the novelty associated with these responses. But it is true that upon initial responding, the novelty and the arising properties of stimuli in the environment could well support some liver pressing. That is true, but not to the extent and for the durations of time we're talking here.
Yeah, great answer, great question. Anna wants to know if the goal is responding and not obtaining the drug reward, then rats would persevere in responding even when responding is no longer reinforced by drug reward. Is this the case?
So that is a good question. But as I tried to emphasize, here the animals are not responding for the drug is only delivered after 15 minutes. And if you omit a drug, then either the behavior is goal-directed or habitual, changes nothing to the fact that the animals would extinguish. Once again, the representation of the outcome is very different to how the presentation of the outcome following the response reinforces the action outcome association or the stimulus response association. Extinction doesn't discriminate between habits and goal-directed behaviors. And one thing that is true, and we haven't done it, we have not challenged these animals to respond longer. We could have seen if we were, instead of training them for 15 minutes, challenge them for 30 minutes. But the fact is, since these experiments were within subject designs, especially for causal manipulations, it could well have been the case that as soon as the animals knew that they went over the 15-minute interval, they would have had there a cue that told them it wasn't a session like every other day. So it's a difficult question to actually fully address.
Interesting. And then Dana says, really great talk. When considering incentive habits, would you argue that even the presence of an inhibitory stimulus is insufficient to suppress this behavior due to the same role of the DLS? And have you looked at this, or are you considering looking at it?
That is a brilliant question. And I will try to be brief. Can you still see my screen? Yes, can you see my screen?
Yeah.
Go.
We have, so let me, once again, I'll try to come back to, we have shown the exact same mechanisms in animals who volitionally stop responding in the face of punishment. Everything I've shown you here was forced abstinence. But in real life, many abstinence events are in the face of punishment. So if you train rats to respond under conditional enforcement here, or habits here.
Wait, we can't see your screen.
Oh, we can see you. I mean, you
can't
have to
see?
share your screen.
Sorry, I'm
Yeah, you have to reshare it. Sorry.
I'm completely stupid. Yes, of course. Can you see it now?
We can see it now. Thank you.
So what we've done was, this was work done by Max Frissac to develop a real model of heroin addiction. Because you can't punish rats under heroin, it's an analgesic. So here, what Max did was to punish the animals in the 15-minute interval period prior to the first heroin infusion. And you can see that rats with an incentive habit or habit, they all decrease responding when they receive punishment. You see that? But there's a big difference, a big difference. If you look at the first interval, so the drug-free responses, when you stop punishing the animals, rats with a habit, they never recover their pre-punishment levels of responding. They do not update. Rats with an incentive habit, as soon as they stop being punished, they go back up. More importantly, and that, if I may, because I think that's important, what Max did was to punish these animals in the last 7 minutes of these schedules, when they're very much engaged in seeking the drug. So the animals learn not to respond during the first part of the interval because there's nothing happening there. What Max discovered, and I think that is profound, is that not only animals with an incentive habit would immediately relapse as soon as the shocks are not presented anymore, but the habit ones wouldn't. But the rats with the incentive habit, they would anticipate the loss of opportunity to respond during shock by increasing their responses in the first half of the interval that is not being punished, but never delivers the drug. So what they're telling you here is, dude, I have to do my responses. If I can't do them when I get punished, I do them before, and as soon as I have the opportunity, once I'm not punished anymore, I'll do them. So that is a brilliant question. We've been testing this hypothesis, and it is indeed the case that incentive habits promote aberrant engagement in responding around the risk of being punished and contingent punishment.
Okay, we have 2 more short questions, short answers, please.
Yeah.
Okay, so then, and this changes gears a little bit, but is the dopamine signal from the nigrostriatal pathway a possible modulator of incentive habits?
Well, science is not about belief. I believe so. The reason is that we've probed these mechanisms using dopamine receptor antagonists in the DLS, right? So all these behaviors are dopamine DLS dependent. And in the DLS, 95% of the innervation is from the nigra. In the PDMS, we are yet to demonstrate that actually we could perhaps influence these responses by manipulating the VTA or the more middle part of the nigra. There must be a dissociation in the populations of dopamine neurons supporting baseline habitual drug seeking and the rebound, this exacerbated relapse that incentive habit support. We are yet to test this hypothesis.
Okay, and then last question, Masaya was asking, can you explain again why the separation of punishment and drug taking is so important?
So,
oh, but also can you explain the difference between compulsive taking and seeking behavior?
Absolutely. So the first thing, this is all literature, the 40s and 50s and 60s. If you take a stimulus that always precedes another stimulus that has itself inherently high rewarding properties, the stimulus that precedes the other one is going to become a conditioned stimulus, right? By definition, a conditioned stimulus acquires the sensory specific and motivational properties of the US. What do I mean? I mean, if you present a light before saccharin, when the animal engages with the light, they may lick the light. If we present the light before food, the animals may chew the light. So there is a sensory specific property there. Now, if that stimulus has itself a aversive motivational value, in other words, it's a shock. If I present a shock immediately before the delivery of food, that shock becomes a CS. By definition, this shock acquires the motivational value of the US. So you can be presenting as many shocks as you want and say, look, it's punishment. No, it's not, because that shock is not a punisher anymore. It's a CS that has appetitive value. And there are a lot of very elegant studies showing this counter conditioning, whereas pairing of a aversive stimulus with a rewarding stimulus in a Pavlovian fashion transforms the aversive stimulus into an appetitive cue. Hence, it's not because you introduce a shock in your procedure that you're measuring punishment. And there are quite a few papers out there where people have shown, oh, there's loss of compulsivity because when I pair the shocks with the US, and they test the animals afterwards, they all resist to shock. No, they're not resisting to shock. They're telling you that they've learned that this shock is not a punisher anymore. It's a CS.
Okay, great. With that answer, we're going to close the session. Thank you so much again, David Belin.
Thank you very much.
It's been a pleasure, and it's been a pleasure to co-host with Sema. Just two more informations before we leave. So all the talks, you can watch them on our YouTube channel. So if it's inconvenient for you to watch it at a given time, you can watch them on your YouTube channel. And the next talk is next Friday by Anna Samaha, and it's going to be hosted by Megan Creed and Ankush Chakraborty. So with that, and I'll let you go, and thank you again.
Thank you very much. Bye-bye.
Thanks. Bye.